The following experiment was on decomposition, and this PowerPoint presentation is done by Catherine Keister. Decomposition is an efficient process in which dead organic matter, such as dead plants or animals, decay. During this decaying process, the organism releases carbon dioxide and other inorganic compounds. Decomposition is ecologically important because the organic matter returns to the soil and the nutrients are recycled and utilized by other organisms in that environment. Here is an image showing what I just said. So leaves fall from the growing tree, and then these leaves die and start to decompose. As they decompose, they release water and carbon dioxide and other nutrients into the soil, and then these nutrients can then be used by growing plants in that same environment. According to Maililo et al., various factors affect the rate of decomposition, such as the chemical quality of the substrate. So how much nitrogen or linen is present in that chemical makeup of that species. The nature of the microorganisms that help in the decomposition process also affect the rate of decomposition. Environmental factors such as moisture or the temperature of the soil also have an effect on the rate of decomposition. The purpose of the following experiment was to compare the rate of decomposition between leaves from 5 different tree species. These 5 species are Fagus sylvatica, commonly known as copper beech, Acer saccharinum, which is commonly known as silver maple, Quercus rubra, which is commonly known as red oak, Cornus stolonifera, which is commonly known as dogwood, and Fraxinus americana, which is commonly known as white ash. The hypothesis was formed based on the chemical composition regarding nitrogen and linen of the different species. It is hypothesized that the Fraxinus americana species will have the fastest rate of decomposition, and the Fagus sylvatica species will have the slowest rate of decomposition. In order to study the rate of decomposition of these plant species, leaves were collected from each of the 5 species. After recording the initial weight of the collected leaves, the litter bag approach was used. In this approach, the leaves were transferred into litter bags, which were sealed using a blowtorch. After labeling the litter bags, they were set in the drying oven in order to remove any added moisture. Leaves were then buried in the Olin greenhouse so that the environment in which the leaves underwent decomposition was similar to that of their original environment in order to obtain the most accurate results. The litter bags were left in the Olin greenhouse for 4 weeks. After this time period, the litter bags were removed from the soil and dusted and cleaned off. The litter bags were thoroughly cleaned in order to prevent any other substance from affecting the new dry weight of the species. The species were then placed in the drying oven once again in order to remove any excess moisture that may be present on the leaves from washing them off. After a few days of drying, the final weight of the leaves were recorded. The first equation shown on this slide was used in order to determine the ratio of dry weight to fresh weight of the leaves. This was found by dividing the dry weight of each leaf species by the fresh weight of each leaf species. Using this value, it was possible to then find the rate of decomposition of each species, which is shown in the second equation. This was found by taking the natural log of the ratio of dry weight to fresh weight, which was found in the previous equation. This equation was then divided by T, which was the amount of weeks that the leaves underwent decomposition. So in this case, it was 4 weeks. These equations were used by 3 to 4 groups in each of the 9 laboratory sections in order to find the rate of decomposition for each species. These values from each group in all 9 sections were averaged in order to find the values used to create Figure 1. Figure 1 compares the average decomposition rates of each tree species. As shown in the graph, the Cornus species on average had the highest decomposition rate, and the Fagus species on average had the lowest decomposition rate. Using Excel software, multiple t-tests were taken between the species with the highest mean and the species with the lowest mean for each laboratory section. The t-statistic, degrees of freedom, and p-value from the t-tests are depicted in Table 1. The t-test indicates if there is a statistically significant difference. If the p-value is below alpha, which in this case was 0.05, the null hypothesis is accepted, and therefore there are no significant differences in the data that would suggest the observed differences were due to random chance. As shown here, the laboratory sections where the data did not occur to random chance were section 20113 and 20111, whereas the other laboratory sections, the p-value was above 0.05, and therefore the null hypothesis was accepted. The species with the highest average decomposition rate and the species with the lowest average decomposition rate in each of these two sections were compared in Figure 2 and Figure 3. As shown in Figure 2, which represents the results from the 20113 section, the Cornus species had the highest rate of decomposition, and the Quercus species had the lowest rate of decomposition. As shown in Figure 3, which represents the results from the 20111 section, the Acer species had the highest rate of decomposition, and the Fagus species had the lowest rate of decomposition. And these were compared using bar graphs, where the species is on the x-axis, and on the y-axis exists the rate of decomposition. So the hypothesis was that the highest rate of decomposition would be the Fraxinus americana species, and the lowest rate of decomposition would be the Fagus sylvatica species. As shown in Figure 1, the Cornus species had the highest rate of decomposition, and the Fagus species had the lowest rate of decomposition. After performing the t-test, the species with the highest and lowest rates of decomposition from the two laboratory sections that were determined to have the most significant data were compared, and these comparisons were then presented in Figure 2 and Figure 3. However, Figure 2 and Figure 3 do not completely support the initial hypothesis. As shown in Figure 2, the species with highest rate of decomposition was the Cornus species, similar to the data represented by the entire class. And as shown in Figure 3, the Acer, or the Fagus sylvatica species, had the lowest rate of decomposition, similar to the data represented by the entire class. It was hypothesized that the Fagus sylvatica species would have the lowest rate of decomposition, and the data collected in this experiment supports this theory. It was hypothesized that the Fraxinus americana species would have the highest rate of decomposition, and this was not supported by the data in this experiment. But instead, the data suggests that the species with the highest rate of decomposition was the Cornus stolonifera species. This experiment analyzed the importance of the decomposition process. This experiment also supported the theory that not all plants decompose at the same rate, and the rate of decomposition is dependent on the chemical composition of the species and the surrounding soil environment. The data from this experiment concluded that the Fagus sylvatica species had the lowest rate of decomposition, and the Cornus stolonifera species had the highest rate of decomposition. The scientific literature that was used for factual information in this slideshow is shown here.